we're going to discuss how to interpret a semen analysis and steps that can be taken to improve your fertility. If you're trying for a baby, the results from a semen analysis can be helpful in identifying specific fertility problems. Semen quality is known to vary greatly between samples, so it's important to produce at least 2 or better 3 of them. Each analysis should be done at least 2 to 3 weeks apart. This will ensure that the sperm cells included in each sample are different each time. This will provide a much more accurate picture of your overall fertility level. You might be wondering where and how you can do a semen analysis. You can buy fertility kits for use at home, but we do not recommend you do this. The results will not be as comprehensive and most likely wrong, because they're not laboratory quality. The best place to do a male fertility test is in a fertility clinic. It should cost no more than the equivalent of about €100. There are several important parts to a semen analysis. The first is measuring how many sperm a man produces in total. Sperm count is determined by how many sperm are present within each milliliter of sperm. A concentration of more than 20 million sperm per milliliter is considered the minimum acceptable sperm count for a fertile man by the World Health Organization. Any figure below this is considered a low sperm count. Next is the quality of the sperm shapes. During semen analysis, sperm is analyzed under a microscope to assess tail, middle, head shape and the proportions. The semen analysis report then often tells you the percentage of abnormal forms. The ideal sperm shape is not produced in high concentrations, so if your percentage of normally shaped sperm is less than 4%, it's considered low by the World Health Organization, and you may find conception especially difficult. The third part is sperm motility, which measures the quality of their forward movement. You're looking for a minimum of 32% progressive forward movement in your swimmers. Fourth is the total semen volume of the sample. This is measured to determine if there is a blockage of the seminal vesicles. Anything below 2 milliliters may indicate an obstruction. Sometimes sperm vitality will be measured. Anything below 60% of living sperm can also suggest an infection, indicated by high levels of white blood cells, often called pus cells. This information is also included in a semen analysis. Even if there are no noticeable signs of infection, it may be wise to investigate and possibly determine a course of antibiotics. pH and fructose levels are also usually measured. Semen with a low pH or the absence of fructose may suggest a blockage within the seminal vesicles, the glands which produce most of the fluid, which ultimately becomes semen. Semen with a high pH could indicate an infection. An ideal pH would be between 7.2 and 7.8. Always keep in mind that many external factors can significantly influence your results. These include diet, physical trauma of the testes, exposure to high temperatures and even radiation, such as the Bluetooth signal from your mobile phone. Oxidative stress is one of the biggest factors for decreased male fertility and DNA damage. This is caused by free radicals in your body, little molecules which interfere with the duplication of the genetic material during the cell division process. Fortunately, this factor can be influenced quite easily with antioxidants. Many scientific studies have conclusively demonstrated that overall sperm quality, i.e. count, shape and movement, can be increased by up to 400% through the consumption of particular antioxidants and micronutrients such as amino acids L-carnitine and L-arginine, and super antioxidants like glutathione or pine bark extract. Fertility specialists therefore highly recommend multi-month nutrient supplementation for all aspiring fathers, because they've been proven successful and cost-effective. These male fertility nutrients are the base therapy for you and your partner to conceive quicker and increase your chances, as well as protecting the previous sperm's DNA from oxidative stress.